move to Emily, who is going to tell us something about individual behavior and how we can maybe begin to answer some of these questions. So over to you, Emily.
Thank you. Thank you, Susan, and thank you to all the organizers. I'm really excited to be here. I've really enjoyed all the talks and discussions so far. And yeah, that actually is a nice segue into what I'll talk about. What I'll talk about is a lot less generative and a lot more observational, at least for now, but I think it does speak to the importance of considering these individual differences as accounting for actually a lot of the variance that we see in brain connectivity. So let me go ahead and share my screen. So the title I had given for this talk was Toward a Stress Test for the Brain. The alternative and slightly more provocative title is it time to put rest to rest? And so I'm very interested in individual differences. And I'd like to start this talk with a quote from Plato, who wrote more than 2 millennia ago, "No two persons are born exactly alike, but each differs from the other in natural endowments, one being suited for one occupation and the other for another." So it's no secret, it's pretty obvious for all of us that live in society that individuals are not identical to one another. We all have our own strengths and weaknesses and idiosyncrasies. And so the question is not so much do these differences exist in cognition and behavior, but the question is, can we measure them and can we characterize them in the brain using the often fairly limited and usually non-invasive, at least in humans, imaging techniques that we have available to us. So one reason to be somewhat optimistic about our ability to capture these at a broad level is that we've observed, and several other groups now have observed, that individual functional connectomes are actually pretty stable within subjects and pretty unique across subjects. And what this looked like in our hands the first time we noticed this result was that if you calculate a whole brain functional connectivity profile for a number of different individuals as they engage in both resting state as well as several different cognitive tasks, it turns out you can actually match with a very high degree of accuracy a connectivity profile acquired from the same person across different days and across different brain states. And so, again, this was somewhat exciting in the sense that there is a good degree of individual information embedded in the connectome that seems to be both stable and unique. Another compelling piece of evidence about the importance of considering individual differences comes from this piece of work using data from the Midnight Scan Club. And this is just a really nice demonstration that once you account for kind of the group level shared blueprint of functional connectivity in the brain, the factor that explains the next most variance is who the person actually is. So the individual factor. And there's also some interesting individual by task interactions. Of course, if we want to know who somebody is, there's better ways to do that than to put them in an MRI scanner and to calculate their functional brain connectivity profile, right? We could just look at them, or we could look at their DNA or their actual fingerprints. So the real interest and the real meat of this work, I think, for myself and many others, is not so much these differences for their own sake, but these differences in functional brain organization in terms of what their consequences are for behavior or how they relate to real world outputs of the brain. And so our group in our initial demonstration of this was able to show that resting state connectivity could predict this trait of fluid intelligence that was nicely mentioned in the last discussion. So this is a trait that varies, even among healthy adults in a normative population. And we were able to show that we could predict this with some degree of accuracy in a fully cross-validated analysis, just using resting state brain connectivity. And we've since extended this approach to show prediction of things like sustained attention and ADHD symptoms, symptoms of autism, personality traits, other cognitive abilities like reading comprehension. So the predictions are far from perfect. I mean, we have a ways to go before these would be rolled out in any real world context, but I think this is a nice confirmation that the uniqueness in the functional brain connectome is also related to meaningful behaviors. And I think one interesting thing that I've noticed over the last few years, especially as the fields of psychology and neuroscience as a whole have sort of had to grapple with the replication crisis and questions about the reliability of the signals that we measure with fMRI, which is certainly an important thing that we're all dealing with now. I've sort of noticed that there's been this focus on, or this assumption, I suppose you could call it, that if we increase individual reliability or fingerprinting or however we want to operationalize it, that will entail an increase in behavior prediction accuracy. And I think that this assumption is wrong, and I will offer some theoretical and empirical evidence to compel that perspective. And so just on the theoretical side, if we think about this proposition that increasing sort of an individual's similarity to him or herself or reliability or fingerprinting will necessarily entail an increase in sensitivity to behavior, we can sort of trivially see that that's not the case. So we could have a theoretical situation where these connectomes are unique but not meaningful. So they could be akin to barcodes where a barcode is unique. It uniquely identifies the item that it is attached to, but there's nothing in the pattern of thick or thin stripes of this barcode that is related to any meaningful feature of the item that it identifies. Conversely, we could have a theoretical situation where connectomes are meaningful but not unique. So in other words, we could have pairs or even groups of participants whose connectomes are quite similar to one another, to the extent that we might even mistake them for each other if we were to do an identification or fingerprinting experiment. But if we also see that these pairs or groups of individuals are similar on some behavioral dimensions, this might actually be meaningful. So this might actually be something that our predictive models could leverage to make better predictions about held out individuals. And so we can pretty easily see that uniqueness does not imply meaningfulness and vice versa. Another piece of theoretical evidence is that, if we think about in the limit, if we were able to identify some signature of individuals that was maximally stable and unchanging, then by definition, we wouldn't be sensitive to the state level variability that might be equally, if not more important than the trait level variability. And so there's several types of state related variability that we might care about. So we might care about things like attentional fluctuations, which can happen on the scale of seconds to minutes. And then there's many other states that we might be interested in. So things like mood, symptom trajectories, response to interventions, developmental and aging time courses. So these are all within subject changes that are happening across multiple timescales. But if we were to optimize our functional connectivity pipelines to find something that's, again, maximally stable and unchanging, then by definition, we're not going to be able to capture interesting state-related changes according to some of these phenomena. There's also a fair bit of empirical evidence now that optimizing reliability does not entail necessarily an increase in behavior prediction. So this, in this piece of work, the authors are directly comparing these two things. And so they're looking, so every single dot in this scatter plot is an edge or connection in the functional connectivity matrix. And what they find is that there's actually very little relationship between how reliable an edge is and how much it's relevant for behavior prediction. So it seems like these two things, the sort of individual reliability and behavioral relevance, might be embedded in distinct parts of the connectome. And speaking even further to that, this is another related preprint from just a couple of months ago, where these authors are showing a broadly similar thing. So here on the left, they're showing the parts of the connectome that do the most work for fingerprinting, so that are most discriminating of individuals. And they're showing the sort of network level interactions that are most important for the fingerprinting side of things. And it turns out that there's actually very little overlap between those edges and the edges that are doing the most work for behavior prediction on these three metrics. So again, suggesting that by optimizing for one, we're not necessarily optimizing for the other. So how do we optimize for behavior prediction, assuming that's our goal? And I personally think it should be. I'm happy to talk more in the discussion about why that is. But this is a basic schematic of what we're trying to do. So we're trying to predict behavior as a function of the brain. And I think there's actually one, so there's all sorts of things we can do on the signal acquisition and the analysis and artifact reduction side, all sorts of very sophisticated techniques to try to acquire better data and clean up our data. And there's been some really amazing efforts on the part of those goals over the last few years. But I think there's actually one really easy way, so to speak, to optimize for behavior prediction. And that's to really strongly consider the brain state or the scan condition that we have people in. So what we're actually asking them to do while they're in the scanner. And so I'll talk a little bit about why I think that's the case. So it turns out that brain state, so really just what subjects are doing while they're lying there, is really can really boost prediction accuracy for cognitive measures. And so in this paper, the authors are showing here in two different data sets, we have the Human Connectome Project here on the left, which is a sample of healthy young adults, and the Penn neurodevelopmental cohort here on the right, which is children and adolescents. And they're using data from the exact same subjects to predict the exact same variable, which is fluid intelligence. And they're just varying the input to the model. So in other words, what task the functional connectivity matrices were calculated on. And you can see that the resting state runs, so down here R1 and R2 in the HCP, and here rest in the PNC, are actually doing by far the worst for explaining variance in fluid intelligence. And you can get a much bigger boost, in some cases, a more than 5-fold increase in variance explained simply by giving the brain something to do. And so it happened that in the HCP data set, the tasks that gave the best prediction of fluid intelligence were the gambling and working memory task. Same thing right here, so working memory and emotion. But even a simple motor finger tapping task gave better predictions of this sophisticated measure of cognition, fluid intelligence, than did rest. And so the way that I interpret this is, so there's also sorts of sort of low-level data quality things that could be going into this. We've ruled out some of those. I think that does actually give you a boost. Again, happy to talk more about that in the Q&A if folks are interested. But the way that I think about this result is tasks are serving to, tasks are kind of a lens through which we project people, right? And so by constraining what the brain is doing at any one time, we're constraining the overall variance across people, but the variance that remains is more meaningful and more stable and more trait-like. And one type of task paradigm that I'm particularly excited about these days is these naturalistic paradigms. So in other words, having people watch movies or listen to stories in the scanner, which is a really rich, dynamic, engaging state for people to be in. And what we find when we do this is similar to what I showed on the previous slide with more traditional tasks. So this is data where these are predictions of, again, cognitive ability based on functional connectivity. And the distributions in purple come from different train test splits where the input is movie watching runs. And the distribution in dark gray comes from resting state runs. And those are all compared to these null distributions in light gray here. And so what you can see is that we get a boost in accuracy by using movie watching, in some cases a dramatic one, as compared to rest. And we also tried predicting socioaffective tendencies from movie and rest data. And these tend to be harder to predict overall. So these are self-report measures as opposed to the behavioral ones in the cognitive battery. And so overall predictions are lower, but to the extent that we can predict this measure at all, we can only predict it from a movie watching run. And perhaps even more interestingly, we find that the specific type of movie or the content in the movie is actually really important for prediction accuracy as well. So those movie watching runs that I showed you on the previous slide were actually comprised of lots of different 3 to 4 minute video clips with varying content. And when we restricted our prediction analysis to connectivity data from each of those clips separately, we found that clips that were higher in social content, as proxied by the number of TRs containing at least one face on screen, were more accurate for predicting this cognitive ability. And it happened that the 3 best performing clips were a clip from The Social Network, Ocean's 11, and Inception. And the worst performing clips were these sort of nature montages or documentaries that didn't have a lot of humans or dialogue in them. And so whether this is a general kind of attention, arousal, or engagement effect, or whether it's really specific to the social content is something that we're still trying to tease out. But I think it really speaks to the importance of considering brain state and even within that, even within a paradigm like movies, the specific content on the screen as a way to really boost sensitivity to behavior differences. And one of the interesting things about movies that you can't do at rest is with movies, you have this ground truth, right? You have a time-locked input that is shared across people. And so it actually has some really nice properties when it comes to denoising. And so if you start thinking about connectivity in the sense of connectivity across brains rather than within brains, you can be much more sure that if you see connectivity across brains during a time-locked movie stimulus, that connectivity is truly neural in origin, because you're a lot less limited by physiological artifacts that mostly occur within brains. And so it has this nice kind of denoising property that lets us more closely isolate signals that we know are neural in origin from the bold signal. And one of the sort of earliest and most elegant, in my mind, ways of analyzing data from these movie watching paradigms is intersubject correlation. So this was pioneered by Uri Hassan back in 2004. And it's this really simple and powerful idea, for those that aren't familiar, that if two subjects are watching the same movie, and if you look at, a single voxel or a single region, the same region in two brains, if that region is showing a correlation across time, then we can infer that that region is somehow responding or involved in processing the stimulus. And what I and some others have been advancing recently is sort of this extension of this technique that we're calling intersubject representational similarity analysis, or intersubject RSA. And the idea is to look, instead of just what's shared generally across all pairs of people, we're interested in looking at regions that respond more reliably in certain pairs or groups of subjects, and how this might relate to similarity of people's trait level behaviors. And so just to schematize that a little bit more, the idea would be that if we take a brain similarity matrix, so just an intersubject correlation matrix from a brain region as people are watching the same movie, and we compare that to a behavioral similarity matrix, we can identify regions where the similarity of the brain time course is somehow echoing people's trait level biases that they're bringing to the table as they're watching this movie. And it turns out that how you actually calculate behavioral similarity is a subtlety that quickly becomes not so subtle. And so there's a lot of ways to do this. And I have like one more data slide here before we can get to questions. But I think this was compelling to me. So how do we actually calculate behavioral similarity between people? I mean, I think the approach that's most common with RSA that would probably occur to many of us first is just the Euclidean distance between two people's behavioral scores. So let's say we have a measure of trait anxiety, for example, and we might hypothesize that if we were to rank our subjects from low to high on this measure, we might see, and this is simulated data, by the way, I wish my real data looked this pretty, but the idea would be that subjects that score similarly on this battery would be close together, or would have more neural similarity as well. So, and this is what that would look like in a two-dimensional embedding space. So it's sort of a relative metric of similarity. So two people that score that are in similar positions, similar relative positions kind of on this the score range would be expected to be more similar in their patterns of brain activity, regardless of whether they score overall low or overall high. But another type of similarity structure that we could think about would be something like this. So for example, in this case, we're predicting that all of the high scorers look similar to one another, but all of the low scorers don't really look similar to one another or to the high scorers. So there's sort of a spreading out, a more idiosyncratic response that's happening at one end of the spectrum versus another. And there's a few other ways that we can kind of calculate these distance matrices that predict slightly different structures and how this looks. But we've been calling these, so we call this model the nearest neighbors model, because it basically just predicts that whoever you're closest to on the scale is the person who you will look most similar to in terms of brain activity. And we call these models the Anna Karenina models, because the intuition is that in this case, all high scorers are alike, while all low scorers are low scoring in their own way. And so it's echoing the famous opening line of that novel, which said all happy families are alike and all unhappy families are unhappy in their own way. And so we do actually see more instances of this Anna Karenina effect in the wild. And so we've seen this now across a few different data sets. So in one study where we had subjects listen to a social narrative that was designed to be deliberately ambiguous, such that people with higher trait paranoia would hear this and become suspicious, whereas people with lower paranoia might get a more neutral or even positive impression of it. We found evidence for this Anna Karenina structure in this left temporal pole region, which is known to be involved in social cognition. And you can kind of see, even in this raw data, that Anna Karenina structure recapitulated. So as we move from low to high trait paranoia in this subject by subject matrix, you can see more similarity, so a more stereotyped response among the higher paranoid participants as they listen to this narrative. Another really interesting example of this comes out of Carolyn Parkinson's lab at UCLA, and they so this is maybe a more interesting sort of real world behavior, getting to your last question, Susan, but this behavioral metric is basically how popular someone is. So they're looking at freshmen in a UCLA dorm, and they're measuring basically the degree of how many connections they have in a social network. And they find that people that are more popular have more similar neural responses during movie watching, while less popular people are more idiosyncratic. So they're not similar to other unpopular people, nor to the high popular people. So that was an interesting one. And then finally, taking it to a psychiatric context of mental illness, there's been a number of interesting studies looking at sort of the typicality of brain activity during movie watching and how that can be used to predict or classify diagnosis. So this is just one example with schizophrenia. You can see these are the raw time courses. You see this nice stereotyped piece of activity here in blue in the healthy controls, and not much stereotyped activity at all in the patients with schizophrenia. So much so that you can use this typicality to actually classify whether someone is a control or a patient. And so you can see this kind of manifesting as all of the healthy controls are kind of clustered here in this part of the space. And the patients with schizophrenia are kind of spreading out in a somewhat consistent direction, but you just see a lot more variance among those individuals. So to summarize, we're seeing that individual connectomes show some degree of stability, but reliability in and of itself, I'm going to argue, is the wrong target. I think we should be optimizing directly for behavior prediction. We're seeing that using tasks over rest can dramatically improve sensitivity to behavior differences, and that's kind of what I mean by a stress test. And we're seeing that naturalistic tasks such as movie watching are particularly promising. There's a lot of outstanding challenges with this work. Hopefully some of them will be brought up in the questions, but some things I didn't talk about. So making sure we're getting prediction for the right reasons. So handling confounds, things that can be collinear with our behaviors of interest. So things like head motion. I think there's a need. A lot of what I showed you today comes from these large end data sets, which are extremely valuable for, discovering and validating some of these brain behavior relationships. But I think we need to balance these with the need for innovation on task paradigms, which can sometimes require smaller data sets, at least to begin with. I think we need better and more valid behaviors, speaking to the point that was brought up at the last discussion. And finally, longitudinal data sets, I think, will really be the gold standard here. A lot of the work that I've done and work that others have done, it's not really prediction. It's a detection of a behavioral measure that was acquired simultaneously with the scan. And I think that's an important proof of concept, but I think ultimately, the value out of this stuff will be if we can actually predict. So say something before it happens. And for that, we need more longitudinal data. And there's some exciting efforts underway to collect that. But, I think we have a ways to go on that front. So with that, I will stop there, and I'm happy to take questions. I think you're muted.
I'm sorry that I keep muting, but we're having a horrible thunderstorm in St. Louis. And I'm trying not to overwhelm the speakers with the sounds of our weather. So thank you, Emily. Thank you for that. And we have a number of very interesting questions that are coming into the chat. So I just wanted to ask you, I'm going to take my chair prerogative again and start by asking you a question, actually just a comment and then a question. So I think it's very true, this question about similarity and how we judge this. In fact, I just took a semester-long philosophy class in which we spent the entire semester talking about where do we make our judgments of similarity from. And so I think that's a real challenge. But I think it's particularly a challenge when we're talking about task behavior, where outputs can look similar, but it doesn't actually mean that the person is performing the same task. Or similarly, we can see that we don't, I think what I'm trying to say is we don't often know what the subject is actually doing. The only thing we can read is the behavioral output, right? So there could be lots of ways that an individual gets to that behavioral output. Is your approach getting at that question? Will we be able to understand the different approaches by which people can reach the same output, the same end state?
Yeah, that's a really good question. I would say, some of the earlier stuff that I showed, we're not really getting at that. I think it's a really important, potentially a really important piece of what's going on. But, I guess when we take these behaviors that are acquired outside of the scanner, these sort of traditional batteries like the fluid intelligence or even some of the, personality questionnaires or self-report measures, maybe it's less relevant for that. I guess it's more relevant for the behavioral measures. But we're sort of taking those scores at face value. So, two people have a score of 10, but that doesn't mean that they use the same strategies to arrive at that score necessarily. And so, I think that's certainly an open question that, that requires more, I guess, in-scanner behaviors, whereas we've usually been looking at these out-of-scanner behaviors that are theoretically more trait-like, but that's debatable in and of itself. But I think maybe, we're seeing hints of, and I think, anyone in psychiatry, I think, would recognize this a lot better than faster and better than I do, just looking at the brain imaging side. But I think there's so much heterogeneity in disease processes and ultimate symptom profiles and things that kind of get lumped into the same diagnosis. And the results I was showing at the end with kind of these models, that it's not, there's not a consistent response in the patients. It's more a spreading of variance, and it's kind of a spreading out around what we consider to be normal or typical. And I think that really challenges a lot of the linear models that we tend to like to use for this kind of a thing. And that suggests sort of a rethinking of just how we kind of rank people and what the dimensionality of the scales should be.
Great, thanks, Emily. Okay, I'm going to turn to Dan, who's this time is going to ask his own question rather than somebody else's. Go ahead, Dan.
Thanks so much for a great talk, Emily. I think this is, as has been hinted at a couple of times today, a really important question about individual differences. And I'm curious on what your thoughts are about using time-varying functional connectivity from REST data as kind of a bridge between the static, kind of reliable individual functional connectomes and these more kind of behaviorally induced brain states, with the idea being that you can get kind of the more individual idiosyncratic brain states from the time-varying functional connectivity, assuming you can do that reliably, but also make use of existing data sets. And there's some work to suggest that, like, as you cited, task-related connectivity predicts behavior better than resting connectivity, that the same is true insofar as time-varying connectivity can predict behavior better than static connectivity.
Yeah, I think that's a good point. And I have a lot. I actually have a opinion piece coming out with the title, "Is it time to put rest to rest?" And I think one of the more promising things, so I think like one of the biggest questions about rest, as you well know, is we see these dynamics at rest, but how do we, is that actually linked to some kind of ongoing cognition or the contents of the mind, or is that due to lower level physiological processes, or just statistical fluctuations around a mean? I'm fairly convinced that there's at least some aspects of cognition that are reflected in the dynamic connectivity even at rest. But I think where we'll get more power is to combine, as I think you're suggesting, combine resting state and task acquisition. So one of the things I think about it, and there's been some really nice work on this from people like Javier Gonzalez Castillo and others, looking at if we can identify sort of motifs or patterns that emerge during task-based connectivity, where we have a ground truth and we know what the person was doing, and then we can kind of detect like echoes or recapitulations of those signatures at rest. It gives us a little bit more of a handle, or a little bit more of a license to make some inferences about what those state-related changes at rest might be actually reflecting from a cognitive perspective, and why we get better prediction accuracy from looking at dynamics at rest. I mean, I guess like one of the big questions about rest that has been asked a lot is, okay, well, you see differences in static or even dynamic functional connectivity at rest, let's say across people or across populations, with and without psychiatric illness, for example. And how do we know that that's really a stable, trait level feature of functional brain organization, or it's just differences in what people are doing while they're in the scanner? So differences in the type of cognition they're engaging in could lead to those systematic differences, and that's not usually being controlled or asked about. So I think also combining rest and dynamic connectivity with some, with, let's say, introspection data. So people like Johnny Smallwood and others who have done really nice work to sort of actually ask, if we want to know what people are doing during rest, let's just ask them. It's not perfect, but I think it gives us more of a handle on that than just kind of going in blind to these resting state signals. And so I think, to sum up, I think dynamics is certainly a very powerful approach. And I think it'll get even more power if we can focus on benchmarking those dynamics with some kind of, ground truth, whether it's from comparing to task data or from acquiring introspection data, acquiring other types of physiological data. So yeah, I'm not sure if I totally.
No, that was great, and I agree with all of those points. I think that's the way to go.
Great, thanks, Dan. Thanks, Emily. Stefanos, returning to you next.
Yes, thank you. So my question actually corresponds quite strongly to what Dan just asked. And it's basically more about graph measures of functional connectivity. So you showed that in the Midnight Scan Club that fluid intelligence is, not the resting state doesn't strongly inform the fluid intelligence. But I was wondering how that would translate to more advanced functional connectivity measures based on graph theory, for example.
Yeah, that's a good question. To be perfectly honest, so I played around a lot with graph theory measures early in my PhD to see if they would get us more power in terms of being able to predict these individual differences. And I didn't necessarily see that was the case. So I think, there's certainly something attractive from the intuitive standpoint about being able to boil down these really complicated matrices into a set of higher order statistics describing kind of the graph level relationships. But I think that there are so many steps, and this is just my personal opinion and what I've kind of seen empirically. I think there is power in these measures for sort of thinking about the brain more generally as a system. But I think when it comes down to looking at individual differences, our signals are pretty subtle. I kind of emphasize the differences in my talk because that's what I'm interested in. But at the end of the day, most healthy neurotypical people are going to be fairly, the group factors, I guess, account for the most variance. And then on top of, the next most thing is the individuals. But, broadly, we all have, a visual cortex in the same spot. We all have a motor cortex in the same spot. We all have a default mode network that tends to, fluctuate up and down and things like that. So the signals that we're picking up on are subtle ones. And, my personal bias has always been to stay as close to the raw data as I can, because I think that gives us the most power, certainly when we're talking about these predictive models. And the graph theory measures, although we've tried them, at least in our hands, haven't really added much to our ability to predict behavior differences. So there have been studies, dating back a while now, some influential studies, looking at how fluid intelligence relates to small world properties of the brain and things like that, and showing compelling correlations within data sets. And I think when it comes to these predictions where you're really trying to generalize out of sample, it can be tricky when you're boiling things down to just a handful of measures for each person that are in turn sensitive to the assumptions that have gone into, how you've calculated those. So how are you defining your network? How are you thresholding your edges? Are you using a weighted approach? And so there's a whole host of analytical choices, even beyond the ones we have to make already to do functional connectivity. And I just empirically haven't seen that add a whole lot in my hands. But certainly others, might be pursuing that approach, and it could potentially add some complementary information.
Great. Okay. Thank you very much, Emily. Thank you, Stefanos. I'm going to turn to a member of our panel right now. Andrea, could you unmute and ask your question? Thank you. If you can turn your camera on, that would be great. If not, we understand.
Yeah. Yes, I can. Hi, Emily. Thank you so much for the great talk. Really enjoyed it. What I found particularly interesting was this Anna Karenina effect that you showed in the in individuals with high paranoid traits during this, in terms of the temporal lobe activation. And then you also showed that there was a reduction in the synchrony of activity in another paper in schizophrenia patients. I guess it was first episode psychosis patients compared to healthy controls. And I found that intriguing because paranoia or persecutory ideation is sort of a core feature psychosis. So I was wondering whether, so how do you kind of reconcile these two sets of results? Could it be that there's different subgroups in the patient population that within that subgroup they are actually more synchronous?
Yeah, that's a really good question. And so full disclosure, I was not an author on that study with the schizophrenia patients that I showed. But I think that's a really good point, because in our paranoia study, we were, and also to be clear, we were looking only at subclinical. So those were all healthy people in that study, but they were just on a range of a trait paranoia measure. And also the stimulus that we used in that study was specifically designed to bring out those differences. So it was a narrative that we actually created ourselves, that was sort of designed to be deliberately suspicion provoking in someone that might be more prone to that type of thinking, and less so in other people. And I can't now recall exactly which movie was used in the schizophrenia study that I mentioned in that same point. But I think your point is a good one. And I wonder if, we were to go back and reanalyze the data from the schizophrenia paper specifically, whether there are potentially subgroups or subclusters of schizophrenia patients that look more similar to one another, and whether that scales with the symptom profiles at all, since obviously it's such a heterogeneous disease. So yeah, it's a great question. I think with more data, with more individuals on the same stimulus, I mean, that's a challenge of these studies. It's hard to sort of aggregate. We want these big ends, similar to some of the large scale data collection efforts that I've been fortunate enough to be a parasite on throughout my career. But there's not as many large end data sets that have movie watching and that have detailed phenotypic characterization, and that also contain both normative populations and patients. So it's a great question. And hopefully, with more data or with finer grained behavioral measures, we'd be able to carve that out a bit more precisely. But I think the last thing I'll mention in response to that is one of the other, cool features of naturalistic tasks, which is maybe obvious, but I still like to point it out, is that when you have this ground truth stimulus, you can actually go back to the task itself. So, you can kind of do a data-driven approach to identify the regions that are more synchronized in, pairs or subgroups of subjects. But then you can actually take that, back to the stimulus itself and ask, okay, well, what was happening to drive activity in those regions that seem to show a difference in their stereotyped activity? So that could be, another way to kind of get at that is, are there differences in the content of the stimulus that might give us a clue as to why we're seeing a relationship in one direction for one stimulus and a relationship in the other direction for the other one?
Yeah, and I think some of this work also echoes what we've also found in individuals with highly paranoid traits compared to first episode psychosis patients, differences in terms of how kind of belief precision drives aberrant behavior. So it might be something to do with the precision of top-down predictions that might make individuals more similar to each other in the sense of how they interpret socially ambiguous information. And related to that, I was also wondering, so I really like these naturalistic studies, and I was wondering, what about using also probing subjects to specifically to make a specific decision. So whether active tasks, sort of active naturalistic tasks, might yield a better, better predictive validity in terms of predicting cognition outside of the task.
Yeah, that's a good question. Something I've thought a lot about is sort of, do we want to introduce behavioral readouts that would be happening during the movie or the story, for example? And I think there's a lot of pros to that, I think you were kind of alluding to. So, sort of having a behavioral readout of similarity to go with our neural readout of similarity. I think the downside of doing that is, it may disrupt the sort of naturalness of the experience, or it may kind of constrain people's cognitions if you're kind of saying like, okay, rate your drag a slider when you're feeling positive or negative about the characters in this story. That would be a really interesting piece of data to acquire, but then of course it might change the experience of the person and sort of cause them to pay more or less attention to certain things, and it might change it for different ways across individuals. So I've struggled with whether or not to do that. I mean, I think in some cases it can be a really powerful thing to do. One thing we have done, which I didn't mention, which we did do in that paranoia study, is we often have people after the fact do a battery designed to characterize their kind of interpretation or appraisal of the stimulus. And we usually start with a free recall task that's just pretty open prompt. And then we can do some really interesting natural language processing things on that to pull out sentiment or, topic models, like choosing, like seeing where people kind of focus their recall or their appraisal. And we can also do sort of more constrained questions about, rating characters on different dimensions. And so that we can get kind of a behavioral similarity profile as evoked by the stimulus itself. And then we can kind of, the ultimate goal is to kind of triangulate between those three things. So you have the intrinsic kind of baseline traits that someone's bringing to the table before they even hear the stimulus. And then you have the patterns of brain activity as they're exposed to the stimulus. And then you have the behavior that the stimulus evokes. So they're kind of state level, what interpretation they actually ultimately arrived at. And there could be relationships sort of between all three of those things. And so usually when we do these studies, we're trying to characterize that as well. And so how to actually operationalize that behavior is still, there's trade-offs, but I think it's certainly a really interesting thing to try to do.
Yeah, I think eye tracking would be really useful to have in combination with those tasks. Thank you so much.
Yeah.
That would be great. Okay, now we're going to thank you, Andrea, for that question, leading the discussion in that direction. Our next question from the attendees is from Samuel Ricom. Please go ahead.
Hello, can you hear me?
Yes, we can hear you fine.
All right, thank you, Dr. Finn. I really enjoyed your talk. My question was actually, honestly, for the most part, answered by your discussion with Dr. Diaconescu. So I thought, like, my initial thought was that psychiatric samples might present a sort of limitation to this approach, just because something like schizophrenia is just such a large syndrome with so much heterogeneity. But actually, upon hearing that conversation, it makes a lot of sense that it can actually work in the sense of dividing the subgroups into like the paranoia group or the other kind of phenotypic aspects of schizophrenia. So I thought that it was mostly answered. So I actually really enjoyed that conversation between the two of you. It's a very interesting approach to it. So thank you.
Yeah, I mean, the one other thing I would say in response to that, also just reading your written comments, is so you can kind of come at it in either a supervised or an unsupervised way, right? So a lot of what I showed in the talk was more supervised. So we have a behavioral metric, that we're essentially hypothesizing is going to show some level of differences in terms of the neural activity across people. But yeah, you could also imagine with enough data, you could kind of subgroup or cluster people bottom up. And the other thing I think is worth pointing out, because I think it's pretty doable data people already have, with clinical populations where you often are kind of boiling people down to their score on a single scale, for example. Yes. One way to get more sensitivity is just to, which is really nicely lends itself to this kind of RSA approach, is instead of taking the total score on a battery, just take the actual vector of item level responses on that battery and use that as your similarity metric across people. So people that, tend to have similar levels on similar questions, so you could have two people with the same total score, but that have, that got to that score in really different ways by having, more positive symptoms or more negative symptoms or what have you. And so there's probably a lot more sensitivity to be had as well by looking at the item level answers to those questions and using that as your similarity instead of, summarizing first and then calculating similarity across the total scores, if that makes sense.
Yes, thank you.
Great. Carl, I'm just going to ask you, is your question or comment a follow-up on the current conversation? If not, I want to go to one of our attendees and then I'll come back to you.
No, go to the attendees.
Okay, great. Thank you. We have a question from Ross, RAS. Ross, could you go ahead and ask your question?
Sure. Thank you, Emily. That was a very interesting presentation. You make quite a compelling case for the role of individual versus group average connectomes. But I wonder, in this discussion of task versus rest and the utility of the task data as opposed to rest, whether there's any consideration for data that's acquired prior to childhood. The two examples you've given were 8 onwards and a period thereafter. But particularly in terms of infancy, and particularly in terms of me thinking of neonatal scenarios, we don't necessarily always have task data, particularly for instance, for term-born babies. I wonder what your thoughts on this would be.
Yeah, that's a good point. You know, I think there's certainly special populations, infants being one of them, and you know, potentially also comatose or patients in a vegetative state being the other one. I will say, you know, I think movies, to the extent that you can do any task in those populations, I think movies are a good candidate. You know, worst comes to worst. I don't do infant imaging, but I have colleagues that do, and I understand it's quite challenging. So it might not be practical with in terms of, you know, the data quality and how you're sort of, you end up kind of concatenating chunks of data when the baby's not moving and things like that. So I don't know how it would interact with the practicalities, but I do think, you know, there's certainly no harm in having a movie playing to an infant. Again, I don't do this type of work, so I'm happy to be challenged on that. And then, you know, to the extent that you do see any kind of synchronized activity, you know, between babies, or you know, perhaps it could be used to actually measure a developmental change in response to stimulation if you had the same infant, you know, scanned multiple times as they develop. You know, it certainly can't hurt. Worst comes to worst, you kind of just treat it like rest. Best case scenario, you might actually find something interesting that's tracking with some feature of the stimulus, even if it's just sort of low level audio visual. I think they do tend to be sleeping, so maybe audio would be better, but yeah, you know, obviously you're not gonna do an end-back task in a baby, but I actually think movies potentially could lend themselves to that in a way that other task paradigms don't. And then it opens the door to more interesting, I'll say, comparisons across age groups, and maybe even across species too. So you know, monkeys can watch movies as well. And it turns out that there's some interspecies correlation that's been shown. But yeah, it's a good question.
Thank you.
Great, thank you very much for that. Okay, Carl, go ahead, please. Thanks for your patience.
Well, no. So yeah, this is a cheeky one, but I noticed in the chat that Susan has actually claimed precedence to putting rest to rest in your opinion title paper. So you're gonna have to sort out some copyright issues here, you know. But with that in mind, with your paper, which I presume is critiquing the use of resting state functional connectivity, you know, as the most efficient way of getting at intersubject variability or psychiatric comparison. Just one very technical issue, which in my world comes up quite a lot, and I think is an important issue in relation to many of the presentations and discussions, is certainly in the context of attentional set and cognitive styles, and particularly in computational psychiatry, is this notion of precision control and excitation inhibition balance that Andrea also mentioned. And when it comes to, if you like, phenotyping that, of finding the right data features that will be sensitive to the fingerprints that reflect variation in intrinsic excitability or the gain or the precision control of the nodes of a graph. It doesn't live in the between node connections, it lives in the within node excitability, which is important to realize because, of course, if you're using functional connectivity, the within node excitability is always 1 because the correlation of activity at that node or that voxel is always 1. So yeah, it's not really a question. I'm going to end with a question, but I'm asking the question really as a suggestion to slip in at the end of your critique of resting state fMRI. There's something profoundly dull-minded about that data feature when it comes to understanding the intrinsic dynamics that reflect, for example, the synaptopathies or the modulatory abnormalities that might explain individual differences. But I'll ask it anyway. How do you accommodate intrinsic differences in coupling and connectivity when using functional connectivity matrices as your chosen data feature?
Yeah, that's a good question. I mean, the short answer is we don't for the moment. Although I guess I'm not clear, and maybe I just missed this, but do you see this as more of a problem for task-based connectivity than for resting state connectivity?
Oh
Or do you
no,
think it's a
no.
problem for?
I mean, I think your arguments about task versus rest, I would imagine in this audience, they're received very warmly. So you need to, from my point of view, old school. What you're talking about is the efficiency of experimental design in revealing the experimental variance in your data that allows you to test your hypotheses or questions. What's the least efficient design you could possibly do? Not actually design the experiment. So, obviously you have to put rest to rest. I think that's a given. What I'm talking about is there are two aspects, I think, to a good experimental design. That's the efficiency of the design itself, over which you have control, but you also have control of the data features. You could log transform, you could use covariance data features and do representational similarity analysis or some covariance decomposition, or you could model the time series. So the data features, whether you're using sort of second order statistics like correlations or variance, as opposed to first order data features like a bold signal or a reaction time, these are all choices you have. When you go to rest to functional connectivity characterizations of fMRI, you're making a strong commitment to particular data features. My question to you is that the right kind of commitment when you know that it's likely that an important amount of intersubject variability may be missed by that kind of data feature?
Yes, I see. Yes, and I completely agree. So, and I feel a little bit self-conscious giving this talk to an audience of people who have been in the field for a while, because I think, it sort of feels like we're coming full circle. Some people never got on the resting state bandwagon to begin with. And to the extent that I've done task-based functional connectivity, at least in recent years and in the work that I showed today, it's really just in service of being able to have a direct comparison with REST and speaking to this issue that you identified of efficiency of design. So, if our goal is to understand individual differences, we get a lot more bang for our buck if we have people doing task than if we have them doing REST. And one of the most straightforward ways to show that is by extracting the same data features on both types of data, which at REST are kind of limited in a lot of ways to functional connectivity. And there's, of course, various flavors of connectivity and ways of extracting intrinsic properties of both regions and connections even at REST. But in some sense, you're limited to some kind of covariance structure in the data itself because you don't have that ground truth of a task. And so to the extent that I've looked at task-based connectivity, it's usually been in service of comparing it to resting state connectivity. But in fact, the direction that I think is more promising is going back to features of actual activation. And so less about the covariance structure, at least initially, kind of getting back to the evoked activity. And I think in ways that are very much informed by the foray into resting state connectivity, which certainly served to break us out of our more traditional GLM based approaches, and certainly served a larger methodological purpose of kind of introducing a lot more sophistication and creativity into the kinds of things that we can pull out of the data and the kind of features we can extract. So certainly taking inspiration from those approaches that were developed on REST, but then going back to task and kind of grounding ourselves a little bit more in first order properties of the data, and getting a better handle on those and how those vary across people before we kind of get back into increasingly higher ordered things. Actually, one slide that I took out for reasons of time was from another group. I can't take credit for this, but also in the Human Connectome Project data, they actually showed that if you just go back to task contrast, so GLM, the contrast between a 2-back and a 0-back task, and you use that contrast map as the input to your predictive model to predict fluid intelligence, you do better than resting state connectivity. So, and that's, again, in the same subjects in the same data set, just looking at, a beta weight map essentially across people. So, that's pretty compelling to me. And it suggests that, maybe we should return to the roots a little bit more, and there's maybe more sensitivity there than we might have thought.
Okay.
Can I make a follow-up comment on that, please, Susan?
Sure, Randy, go ahead.
I just want to see if I can clarify my understanding of what Carl's question was and what Emily's response was. My understanding, Carl, is it's sort of saying that if you're looking at only the second order statistics of the signal, i.e. the correlation that you might miss with the local dynamics you're doing, which would be the mean. Now, I guess you could probably include both of those in the term. And I know that, for example, some variations of DCM do that. So you can actually model both the coupling term between populations as well as the mean levels of the populations within a given region of interest as well. And I guess the assertion is that it's specifically in populations where they got a psychiatric issue, for instance, where there might be a change in excitability, by being able to model that, you may get additional predictive power. Am I capturing that correctly?
Yeah, absolutely. I think Petra's here somewhere, and she, with all the virtual brain people, all the work you've been doing, would speak to this notion of intrinsic excitability of neural populations as being probably the most important shaper of the functional connectome. But just to connect it back to Emily's answer, I think, in a sense, you actually just also made the same point when you were talking about, having the ability to use evoked responses to get at these changes in gain, if you like. So from an old school perspective, you're just talking about a factorial design. You're talking about a design where you use one factor to evoke the response and another factor to see whether that response was amplified or attenuated in a different context. And it's that sort of second order, it's that beyond the correlation, if you like. So again, you could only, I think, do this reasonably to get at this intrinsic excitability aspect. Almost by definition, you'd have to have a factorial design, which again precludes resting state studies because there's only one factor, which is time. Unless, well, you could argue dynamic connectivity would provide another factor, but I'm very pleased to if there is an opinion piece coming out where you are closing the circle and going back to basic principles, there are lots of really powerful arguments to motivate that along the lines of exactly what Randy was talking about. If you want to get at the excitability thing, you have to design the experiment to do that.
Completely agree.
Yeah. Emily, we're going to close out with a comment from one of the participants, but then we're going to ask Rory Boyle to ask his question. Both of them were interested in, so this might be a bit of an easier question than Carl's for you to answer, but it has to do about introduction of physiological artifacts into the data. So for instance, heart rate or other kinds of things. Why don't you go ahead, Rory, and ask your question.
So hi, Emily, thanks very much for the really interesting talk. And I was just wondering if you were to use movie watching paradigms over resting state paradigms, there might be a risk of introducing sort of quite subtle motion related artifacts due to the increase in eye movements. And would there be any, if so, would there be any sort of fairly straightforward ways of controlling for this if you don't have eye tracking software within your scanner?
Yeah, that's a good question. Although I will say that one of the other advantages of movies that I didn't show is they actually they tend to reduce head motion overall. So just sort of global head motion. They actually increase subject compliance, especially in hard to scan populations like children or psychiatric patients. And that's maybe just because rest is pretty hard to sustain. It's pretty boring. Turns out that if you give people a movie to watch, they're more content and they'll lie more still overall. I think the point about eye movements is a really good one. There's not as many simultaneous eye tracking, fMRI movie watching data sets out there as one might like. I've seen people do a few different things for this. So actually there's some really amazing deep learning techniques now where you can actually recover eye gaze position from bold signal in the eyeballs. So that's one approach. If that's in your field of view, you can actually recover where people were looking at a TR by TR basis by running that through this neural network, which is pretty cool. Another thing I've seen people do is take the signal from the frontal eye fields and try to just sort of correlate that across subjects and see or regress that out in various ways. I think it is in some ways an open question, how much of these differences are driven by differences in where people are looking, although I would argue that's my perspective on that is not necessarily an artifact. I mean, so it's a bidirectional relationship. So what's on your retina is going to affect your brain activity, but also, your top-down attention processes and, your yeah, just where you're choosing to attend, you know, is arising from your brain, and that's going to affect where your eyes are going. So, you know, it may be that eye movements account for some of the differences in synchrony that we see. I don't necessarily see that as an artifact in of itself. I do think it's important to explore, you know, how much variance is due to that. And so, you know, hopefully as we get, you know, there have been some studies, I think people usually find that it explains some variance, but not all. But we actually just got a brand new MR compatible eye tracker here at Dartmouth. So I'm excited to start looking into that more.
Great, thanks very much. Thank you.
Okay, and with that, I'm afraid, Emily, that we're out of time. But I think, again, thank you so much for your talk, stimulating a really wonderful conversation. We'll give you a round of virtual applause.